All right, so let's go ahead and get started. Thank you everyone for joining our workshop today, which will be on utilizing t-SNE for flow cytometry. Our workshop will be run by Robert Ladd, who is a facility manager at Loyola Medical Center Flow Cytometry Core. Before I hand it off to Bert, I just want to share a few company slides to introduce Proteintech. So, Proteintech is a leading manufacturer of antibodies, proteins, and immunoassays across several different research areas. So, our portfolio comprises of three core product lines. So, we have our Proteintech branded antibodies and immunoassays. So, we have a very expansive portfolio of antibodies that cover over 13,000 targets. We also offer antibody labeling kits, ELISA kits, IHC kits, and cell separation kits. We also have our humankind branded bioactive proteins, and all our proteins are expressed in human HEC293 cells so that you can be confident that they have native folding, post-translational modifications, and glycosylation. And then lastly, we also have our Chromatech branded nanobody-based reagents, which are based on alpaca nanobodies. And due to their small size, they're really ideal for clean immunoprecipitations, immunofluorescence, and protein purification. So all our product lines share a common value under the main Proteintech brand, so you can be confident they are of high quality and validated products, but they each address its customers in a separate and specialized manner. I also just want to take a minute to celebrate the success of our customers. So Proteintech is really proud that our products have been cited in over 200,000 publications worldwide. And we've also were the Researcher's Choice Award winner last year from CiteAb. So we're really proud of that because it shows that our customers really do value our products. So with that, I'd like to go ahead and introduce our speaker for today, Robert Ladd. So Bert is the facility manager at the Loyola Medical Center Flow Cytometry Core. He received his bachelor's in biology at UNC Chapel Hill and worked in the University of Chicago CAT facility as a flow cytometry specialist before transitioning to Loyola to run his own core facility. His passion is flow cytometry and equipping his user base to use the technology to its fullest potential. So with that, I will go ahead and I will pass it off to Bert. If you do have any questions for him during his talk, please input those into the chat or the Q&A box, and we'll address them at the end. So thank you so much, Bert, for being here, and I will go ahead and pass it off to you.
Yeah. Thank you very much. Let me get this up. All right, so thank you everyone for tuning in today. As was said, my name is Robert Ladd. You can call me Bert. I am the facility manager at the Loyola Flow Core, as was said. Today I'm here to talk about t-SNE, specifically the theory behind it and the practical steps of actually using it on your data. The reason I wanted to give this talk is because t-SNE is increasingly becoming a very common, almost obligatory part of analyzing any high parameter flow cytometry data. But unfortunately, despite its popularity, it is not a one and done method. It's not very plug and play, you might say. It can require some trial and error, some optimization. And doing this without understanding what's actually happening under the hood can be really frustrating. You're kind of stumbling around in the dark, just turning knobs at random. You're not sure what to do. Like, what is perplexity? So what I hope is that at the end of today, you'll have a better understanding of what's going on fundamentally with t-SNE. And using that, you'll have a better idea of not only how to optimize it, but also to interpret the output. In terms of scope, I am going to be focusing on t-SNE. There are other dimensionality reduction or clustering methods out there, some of which are very similar, but that would be a much longer talk. So we'll be focusing on t-SNE. And when relevant, this will be in the context of flow cytometry. Again, a lot of this information will apply generally to t-SNE no matter the application. But when in doubt, I'm going to defer to my expertise, which is flow cytometry. So with that said, let's jump into a little bit of background on t-SNE. Let's go ahead and get this out of the way first. How do you even pronounce this? Some people say t-SNE, some people say t-SNE. t-SNE is probably the more accurate one, but unfortunately for you, I think t-SNE sounds more fun. It rolls off the tongue easier. And if I say t-SNE, then my tis the sneezing joke on the first slide doesn't really work. So I'll be saying t-SNE for today. In terms of what it's actually doing, it is a dimensionality reduction method. In very simple words, what that means is we are projecting or arranging our high dimensional data, our n-dimensional data, onto a lower dimensional space, typically 2D, although you can really do whatever sort of representation you want. You can think of a shadow as a very simple example of this. We have some high dimensional object, a hand, for example, that is being projected onto a wall, and we see a two-dimensional image of that. We can still tell something about that high dimensional or that three-dimensional object based on the shape of the shadow, but we do lose some information by reducing that third dimension away. For dimensionality reduction to be very useful for data analysis, we need to preserve something about, ideally as much as we can, about those high dimensional relationships. Taking this plot I have here on the right, a very simple CD4, CD8 plot from T cells, I could reduce this plot very easily simply by taking my y-axis away and just looking at all my dots squished against the bottom there. However, in that case, I've not preserved anything about this higher dimensional, in this case, two-dimensional data, so that is not going to be optimal dimensionality reduction. To give you a very simple sort of analogy for what tSNE is doing, and then we'll dig in deeper. Again, looking at this plot here, especially that population in the bottom right, I could say that in my y dimension or my y-axis, all of these dots are similar in that they are low intensity on that axis. Similarly, on my x dimension or my x-axis, they are similar in that they are relatively high values. So I can draw a gate around this, and you could say that I've clustered this data together. In a very simplified sense, that's kind of what tSNE is doing, but rather than just looking at the two-dimensional space here, it's looking at however many dimensions in your data you have, 20, 30, 40, mass cytometry, maybe even more. Why it's useful, our output of a tSNE is going to look like something on the left here. This is one of your prototypical tSNE representations you'll see. The idea is that the data points within each of these clusters have some level of high dimensional similarity. So if we were to look at all of the possible parameters that we could normally plot, dots within the same cluster are going to be very similar in some or most of those parameters. We have some limitations of typical flow data presentation that this can help us get around. With flow data, we can easily plot data in the 1 dimension, 2 dimensions, even 3 dimensions. And if we really wanted to go crazy, we could go further. We could have a 3-dimensional plot where every dot has its own color or shape or opacity, and we can add dimensions in that way. But very quickly, we end up with just a big Picasso-looking mess that's really hard to interpret, and we might as well have just stuck to the 2D plots. So tSNE gives us a way for our simple monkey brains to look at this high-dimensional data and see something we can more intuitively grasp, these high-dimensional relationships. It is primarily a visualization tool. Do not think of it as a way to completely replace all other analysis. You're not going to be running Flow, running a tSNE on it, and sending that off to nature. It's not going to get you anywhere. In fact, you should be using your other analysis tools, your conventional analysis tools, to reinforce and validate your tSNE. Just as a simple example, if I'm doing an immunophenotyping panel and I do my traditional gating for all my subsets I expect to see there, and they broadly overlap with what I see in my tSNE, I can feel a little more confident that I'm actually seeing something real for my tSNE. It's actually representing these high dimensional relationships. tSNE scales up well with large data sets. That does not mean it is fast, quite the opposite. It can be quite a slow algorithm if you just plug in millions and millions of data points. But what I mean is that tSNE doesn't really care how large or how dimensional your data set is. It might take longer to run. You might not get the best separation. We'll talk about ways to mitigate that, but tSNE is a very flexible algorithm. And it can be really useful for what I call data expeditions. Some people call it fishing. The idea is that with traditional flow analysis, we tend to fall into very binary ways of thinking. Is this positive or negative expression? We might get a little more granular and look at low, intermediate, and high. But regardless, we're being very discrete here. And biology tends to be more continuous. So there's some subtleties there that we can miss that tSNE can help us identify. It is, of course, not a perfect algorithm. If it was, we would need to talk on it. Some of its weaknesses we should talk about up top. It's not a very quantitative method. When we get to the math, you might think that's silly. But what I mean by that is I often get the question, I have this plot where it's tSNE 1 versus tSNE 2. What do these axes actually mean? And the short answer is they don't really mean anything. They're really just the distribution along which tSNE has clustered your data so that they are separated. And that's why it's really a visualization tool. Furthermore, tSNE doesn't do a great job of preserving the global structure of your data. What I mean is that 2 dots or 2 data points being in one cluster tells us something about their high dimensional similarity. But the fact that two clusters are next to each other doesn't really tell us anything about the intra-cluster similarity. There are other algorithms, UMAP, for example, that does a better job of this. But for t-SNE, we're really focusing on what gets clustered together. It is a stochastic algorithm. That is the S in the SNE. What that means is that when we first put our data in the low dimensional space, it's going to be a little bit different every time. That can be a little hard to wrap your mind around at first. Makes troubleshooting it and optimizing it sometimes a little frustrating, but it's something you'll get used to, and we'll see why that happens. It's also non-parametric. What I mean is that if I run a t-SNE on 10 samples today, and then tomorrow I run a new sample and I want to add that to my pre-existing t-SNE, I can't do that. I'm going to have to rerun my t-SNE. There's not a way to just plug it in, at least not without a lot more work. As I said, it is a very computationally intensive algorithm. It can take a long time, especially with large data sets. We have ways to mitigate this with downsampling and some derivatives of t-SNE. We'll talk about those towards the end. But regardless, it can take a while, and you're going to have to be comfortable with tinkering with the parameters. Perplexity, iterations, these things can often be kind of confusing for people getting started with t-SNE. But by understanding the fundamentals of what's happening, we can be much more intentional with how we tweak these when we do our runs. So with that out of the way, let's get into the theory a little bit. This is pretty much all you need to know. We got our main equations on the left here, and we plug that into the algorithm. And I'll give everyone a second to write that down. And that's pretty much it. So if anyone has any questions now, I'll take those. No, I'm just kidding. So it's good to know the math behind t-SNE, but what I really want to do today is break it down into a more conceptual level to give you kind of intuitive understanding of what's happening. So I'm breaking it down into three main steps here. We're going to, in the high dimensional space, our original data set, we're going to measure distance between points and assign similarity scores based on that. We'll then project our high dimensional data onto a low dimensional space. And then that low dimensional distribution is going to be iteratively adjusted until we have something that accurately represents our original high dimensional data set. So 3 steps, and then our goal at the bottom there. To use an example, we're going to have a very, very simple data set. This is just a 2-dimensional data set that we're going to reduce down to 1 dimension. With normal t-SNE, you'll be doing something like 20 dimensions down to 2, but the principles are exactly the same. This is just so that we have something very concrete that we can point to. Admittedly, I've kind of biased us already. I've color-coded these by their obvious clusters, and it is a very small data set, but again, the principles will be the same regardless of the scale. So our first step is going to be finding and calculating these high dimensional similarities based on their distance. What that looks like, using our 2-dimensional data as an example, we are going to start with some initial point, in this case, that first red dot, and we're going to measure the raw distance to every other point in the data set. In this case, I'm just showing 2 as an example, 1 red near neighbor and 1 blue not neighbor, but we will do this for every single point and in both directions. So A to B and B to A for every single point. Once we have these raw distances, we're going to plot them under a normal distribution like this, where it's centered over our initial point. So that first red dot right there. The distance of any neighbor or not neighbor to the center of the curve is going to be based on that raw distance that we found over in our 2-dimensional data set. And then from there, we draw a line up to the curve. The closer to the middle you are, the taller the curve is going to be at that point. And the length of that is going to be our similarity score. So you can see the closer to the center, our more similar reds are going to have a higher similarity score as opposed to the lower similarity score of our blue dot. Full disclosure, I basically made these numbers up right here. It's just as an example, but the principle is the same. The closer to the initial dot you are, the higher your similarity score. The end result, and what's really important from this, is we get a similarity matrix that looks something like this. So all of our points by every other point and n by n, essentially. And we have these green cells indicating very high similarity and these orange cells indicating lower similarity. You'll notice the gray diagonal through the middle there. That's every dot's or point similarity to itself. As far as t-SNE is concerned, that is a 0. Doesn't really matter to us. A point similarity to itself should be perfect. We don't really need to know the exact details of that. But from this high dimensional data, we now have this similarity matrix. And this is going to be the gold standard. Once we put this into our low dimensional space onto our t-SNE plot, how similar this is how similar that new matrix is to this one is going to tell us how well our t-SNE has performed. And we'll see that in just a second when we project our data down. So as I said, a very simple example to start. We're going to take our two-dimensional data and randomly, keyword randomly, project it onto a one-dimensional line here. The random is where that stochastic nature comes in. And you can see now we have points that are just totally in a messed up order. However, at this point, we're going to do very similar steps to our last step. We're going to measure the raw distance from our points to all the other points in our data set. So just using the same 3 as an example, we have our initial red point in the middle. Its near red neighbor is now much further away on this low dimensional space, and that blue not neighbor is now very much closer. Once again, using these raw distances, we're going to plot them under a curve. Critically, though, this time we were using a t distribution rather than a normal distribution. This is the t in t-SNE. The reason we use a t distribution here, I won't get too much into the nitty-gritty, but essentially t distributions have a shorter peak and fatter tails or taller tails. And what that lets us do is if we have a point that is very, very far away from our initial point compared to a point that's somewhat far away, with a normal distribution, they will both basically be 0. The t lets us get a little more better separation between them. This is what we call the crowding problem. So that's why we use a t distribution here. The end result is somewhat similar. We now get a new low dimensional similarity matrix, but since our initial order is random, our similarity matrix is totally scrambled. We've kind of lost all that nice perfect diagonals we had in our high dimensional similarity index, or matrix rather. So the next step now is to fix it. We're going to take that low dimensional distribution and we're going to adjust it until we have something that accurately represents our high dimensional data set. So starting with our random initial order and the scrambled matrix that we derived from it, we're going to iterate. This is the iterations parameter you've maybe seen. And with each iteration, very similar points in the high dimensional space are going to attract each other. Very dissimilar points are going to repel each other. So over time, we will end up with something like this, where we now have all of our points clustered very well based on their high dimensional similarity. And if we take this bottom low dimensional iteration and make a new similarity matrix, we have something that looks just like our high dimensional similarity matrix. This is how we know we've reached the endpoint of our t-SNE, and we have something that accurately represents our high dimensional data. The key variable here is something called Kullback-Leibler divergence, or KL divergence. You can think of it very simply as just the difference between two probability distributions, or in this case, difference between our two matrices here. Once we have minimized that divergence, once our low dimensional matrix looks like our high dimensional matrix, aka we have that nice diagonal there, we know that we've reached an endpoint of our t-SNE, and we have something that is going to be useful for visualization. And that indeed is our end goal here. So just as a quick overview of all those steps, two-dimensional data with or high-dimensional data with a high-dimensional similarity index. We project that onto a lower dimensional distribution and therefore get a scrambled similarity index. And then we iterate that, adjusting so that similar points attract, dissimilar points repel, until we end up with a new similarity matrix reflecting our original one. Now that we have talked about the nitty-gritty going on behind the scenes, I want to take some time to talk about the actual practical steps towards setting up your t-SNE. It is not as simple as taking your data file, clicking the t-SNE plugin button, and letting it go. You can certainly do that, but you're probably not going to get very good results. So let's talk about some steps to make that more fruitful for you. I break this up again into 3 main steps: pre-processing, processing, and then executing the actual algorithm. I'm going to do my best here to keep this very general. I don't want to be biased towards any one platform, FlowJo, Cytobank, etc. But I will be defaulting to FlowJo if I need a specific example, just because it's what I know best. Furthermore, this is not meant to be a hard step-by-step protocol for the best way to run your t-SNE. I am trying to keep this general. But at the very least, for each step we discuss here, you should think about why you might do this step or why you might skip this step. And that will give you just a better understanding of your data and ultimately a better output from your t-SNE. So starting with some pre-processing, this is not going to be anything too exotic. We're just going to do some simple gating to really nail down our actual population of interest. So in this case, just a simple example, gating on our cells and forward and side scatter getting on our single cells. If we have a viability and or dump channel, we should go ahead and take the negative from there. We don't want to cluster all of our dead cells. And then from there, it's really up to you. If you want to go specifically into CD45 positive, like I've done here, you can do that. If you want to focus specifically on T cell subsets, you can go to your CD3s, really whatever is going to be relevant for what you care about clustering. As far as some less obvious pitfalls that I've seen people fall into, we have things like antibody aggregates. If you've done any amount of flow or a fair amount of flow, you've probably seen something like this in the bottom left. We have our very obvious cell populations, a double negative, a double positive, a single positive, but then we just have this strange spike kind of going off into infinity, and then we get these weird artifacts where it gets really, really bright and saturating our detectors. Almost always what this is are antibodies that have precipitated out of solution, and now you have these aggregates. They are incredibly fluorescent. They don't really look like cells, and in fact, t-SNE doesn't know that they're not cells, so it's probably going to cluster them together, and you're going to get what I call a phantom cluster. We can gate these out, although you can see in this example, they're typically not a very large population, so if you don't gate them out, it's not going to be the end of the world. This is just something we can do to clean it up. Furthermore, we need to take autofluorescence into account. Depending on the platform you're using or the instrument you ran your data on, you might not have a ton of options for dealing with autofluorescence, but at the very least, we should be aware of it. If you ever plot data like this and you see this perfect 45-degree angle spike off into positivity, you should be skeptical. There are platforms, some spectral cytometers, for example, like the Aurora, that allow us to extract autofluorescence. That can be very useful for cleaning up our data before we run our t-SNE. Another very important step, and frankly, this is something you should always do with your flow data, not just for t-SNEs, is looking at our data on a time axis. So these are two very, very obvious examples, but it can sometimes be more subtle. We have what we should be seeing is a perfect straight smear across the time axis. But in this case, we have some kind of striations, little discontinuities in our data. Almost certainly, this is just unstable fluidics, little clogs that form and then get pushed through very immediately. The way to deal with these is ultimately at the sample prep stage. If you improve your digestion, if you run your cells through a strainer right before you run them, having a very clean instrument, although we don't always have the most control over that, especially in a core facility. But just doing a little bit of cleaning, doing a little bit of better prep beforehand can help you deal with this. However, if you're at this point where you've already run the data, you can't go back in time and just fix the prep. So instead, we have some tools to help us deal with this. Two I'll highlight here are Flow AI and Flow Clean. These are scripts that are available in FlowJo as plugins, for example. Essentially, they're going to look at your data on the time axis, and you have some other tunable parameters there, and it's going to excise those regions of your data that are what we'll call cloggy stretches. After that, you'll get two populations of, quote, good data and bad data, and usually that's going to give you much better t-SNE performance. The reason this is so important, kind of like with our antibody aggregates, if we leave these cloggy stretches in, they are often going to be not very similar to our actual cells that ran normally, but they are going to be very similar to each other. Sometimes if we look at them on a fluorescent axis, they'll be very high or very negative in terms of fluorescence. Excuse me. So t-SNE will tend to cluster those together. And again, we'll get one of those kind of phantom clusters there. I can think of at least 2 or 3 examples off the top of my head. I've had a user come to me very excited about this novel cluster they found in their t-SNE. They looking at all the expression, it doesn't seem like any known cell population, but almost always we look at it on a time axis and deflate their dreams a little bit. So really a very important step to take before you actually run your algorithm. And again, really for any flow analysis, you should do this. Now that we've done the boring cleaning up top, we can do a little more processing before we actually run our algorithm. We should go ahead and trim our files down. That's the gating we mentioned earlier. And I really recommend you go ahead and export these into a new workspace. It's not strictly necessary, but it's going to make it much easier if you don't have to deal with your trimmed files and your raw files that are just massive and have all this debris and stuff in them. Furthermore, at this point, we should go ahead and zero in on our compensated parameters or our unmixed parameters if we're using spectral cytometry. This is really important for a couple reasons. First of all, there's really no reason to run t-SNE on uncompensated data. It's not going to be representative of the true biology that you're interested in. And also, very important. If we're looking at samples from different days, we should have run fresh compensation controls on each of those days, and we might have slightly varying compensation matrices between those days. We really want to make sure that every sample has the appropriate compensation applied to it, and that we're not trying to use some one-size-fits-all compensation matrix. You're just going to be lying to yourself if you do that. So this is just another good practice for your compensation and data. We also are definitely going to need to downsample our data unless you ran very small files. This is mainly a practical concern. If we run a t-SNE on something that has millions and millions of events, it could take days, and you might not even really get that good of a separation, which is very disappointing when you've been waiting a very long time for it. So we need to have that trade-off of size versus time. We do want to give equal weight to all of our samples. So our end result here is we're going to trim, downsample, and then merge all of our samples together, and then we'll run a t-SNE on them in aggregate. We want to make sure that all of our samples we're putting together are given equal weight. So we don't want to have some samples that have 50,000 events and some that only have 10,000, because then those former samples are going to be overrepresented when we do our clustering. Typically, I'll just base it on my sample that had the least amount of events, and I'll just apply however much I can get out of that. I'll downsample the rest of my samples to that level. We also want to make sure, and this is why we merge all of our files together, that every possible population, every possible cluster is represented in our merged file. If we don't do this, then we might have some very rare, very interesting population in our data that we're not going to be able to actually see in our t-SNE. And it's very important that we can actually find those. And so then the last step is going to be actual concatenation. That is going to be the merging of all of our data files together. At this point, we're just going to have one big data file, but we'll still be able to pick out our individual samples. The easiest way to do this is just looking at your sample ID. You can actually, put this on a histogram essentially and gate on your individual samples. You can also, if you're feeling brave, do some keywords, and these are a very good thing to get comfortable with. We could break up our samples by tissue, by mouse ID, by mouse ID, by time point, anything that's going to be most relevant to our analysis. And now that we've done all that boring cleaning, it's time to actually run our algorithm. So we have a couple of decisions to make up top before we actually click go. First is deciding on the relevant parameters to cluster our data. Usually these are going to be fluorescent parameters, but it's not as easy as just picking every fluorescent parameter you ran and saying cluster on these. You should really focus on which parameters are going to actually separate your data. If I have a parameter that all of my events of interest are going to be positive for, there's no real reason clustering on that, because there's not going to be any high dimensional difference that I'm going to find interesting in my clusters. Furthermore, any of the parameters that you already took care of in your pre-processing, live dead, dump channels, for example, there's no reason to include these in your clustering parameters. You've already taken the relevant portion of those, so you're really just going to be getting nothing additional out of that when you run your t-SNE. And then a question I get a lot is, should I include my scatter parameters, my forward and side scatter, when I run my t-SNE? And this is very much going to be dependent on your actual application. If we're looking at something very homogeneous in terms of morphology, just like a cell line or something like that, the scatter parameters typically aren't going to give you too much information, especially if you've gated out your dead cells and things like that. On the other hand, if we're looking at something very heterogeneous, where we maybe care about size, maybe we're looking at development of certain cells or something like that, scatter can add some really important power to our t-SNE. So again, just going to be very dependent on what you actually want out of this dimensionality reduction. And then one of the real headaches for people who are new to it are tunable parameters, sometimes called hyperparameters. Perplexity, iteration, and learning rate are the ones I'm going to focus on here. Perplexity is the big one, appropriately named because it can often be very perplexing. The way I would encourage you to think about this is it is your best guess at the number of high dimensional neighbors you expect each point to have. So generally speaking, if I increase my perplexity for each point, t-SNE is going to consider more nearby points as possible neighbors that could possibly be clustered with that point. It's also, I kind of skipped over this earlier, but the higher your perplexity is going to lead to broader normal curves when we're first figuring out that similarity score. That's just how it's incorporated into the math there. Generally speaking, A higher perplexity, especially for very complex data, is going to give you more separated clusters. But because it is considering more possible neighbors for every point, it is also going to increase the time or your runtime of your algorithm. There is no concrete equation to figure out exactly what your perplexity should be. It really works for a very large range of datas, like 5 to 50. Usually I will, depending on the complexity of my data, I'll start around 25 to 30. And if I'm not getting good separation from my clusters, or in the case where I'm getting too much, I might increase my perplexity and try again from there. This is why downsampling your data so runs don't take as long can be really valuable. Once you found your perfect parameters, maybe you increase the size of that merged file to run your t-SNE on. Next is our iteration. This is simply the number of times it tries to adjust your low dimensional distribution so that you get something that accurately represents your high dimensional data. Once again, more iterations is typically better, but it is of course going to mean a much longer time for your algorithm. This is where the meat of the computation comes in. The more we do iterations, the lower that KL divergence is going to be, and therefore the more similar to our high dimensional data our low dimensional matrix is going to be. Last is the learning rate. This one I typically don't fiddle with too much. The basic way to think about it is the larger your learning rate, the bigger the steps between iteration, the bigger swings the algorithm is going to take in terms of clustering your data. Common knowledge or sort of conventional wisdom is to set this at somewhere between 5 to 10% of your total input events. I usually go towards the higher end of that. But again, not one I spend too much time fiddling with. The important thing about it is that if our learning rate is too low, then our algorithm can get kind of stuck in these local minimums, and it's not going to find the true best representation of our high dimensional data. So finding these best values on your own can be pretty frustrating, but their help does exist. One I want to highlight is the tSNE. This is a t-SNE derivative that keeps a very close eye on that KL divergence, so the dissimilarity between your low and high dimensional representations. The tSNE is going to keep an eye on that value, and once it has been sufficiently minimized, it knows that we've reached the endpoint of our t-SNE. In terms of how many iterations it's going to run. It will also control something called early exaggeration. I didn't mention this earlier for the sake of simplicity, but early exaggeration essentially is in the early steps of our t-SNE. We take really big swings, we multiply everything by a bigger number, so we get these really big jumps between our iterations. Once we have reached a point where kind of our base clusters have been formed, we can take off that early exaggeration and start to fine tune our cluster borders. So tSNE takes care of this for you. I really like tSNE. It takes a lot of the guesswork out of this. There are also some options for just speeding up the t-SNE. Just a couple of examples here. There are GPU accelerated t-SNEs, usually just runs on the CPU. t-SNE CUDA and Cytobank as an example. There's the fast Fourier transform version that I haven't really played around with, but just another way to speed up the computation. And then depending on your platform, cloud computing might be available. You don't have to wait on your own wimpy PC to run this calculation. You can borrow some beefy PCs from the cloud. Cytobank, for example, has this as an option. And I would be remiss if I didn't mention UMAP. It is a faster algorithm. In terms of what's actually happening fundamentally, it is very similar to t-SNE. A couple of fine mathematical details are different. But it does do a better job of preserving the global structure. So what I mean by that is in t-SNE, as I said, two clusters being next to each other doesn't necessarily tell us anything about the intercluster similarity. With the UMAP, that's not the case. It can tell us something more about that global structure. So just last bit here, I wanted to have a few notes on interpretation. This is a tough part to do because it's going to be very specific to what your actual experiment is. But just to give you some general best practices, we really do want to avoid what I call some mirages, kind of like those phantom clusters where t-SNE can make it seem like there's something significant there that maybe doesn't really exist in your data. The big ones being the cluster to cluster difference, which I've already talked about. Also, the actual size of your individual clusters doesn't necessarily tell you much about the clusters themselves. What we really care about is which events get clustered to which events, and therefore our neighbors in that high dimensional space. We don't have to do this alone. We have some co-pilots that we can recruit to come along with us. There are ways that we can validate our t-SNE performance. Just because we get nice separated clusters doesn't necessarily mean it worked perfectly. We should dig in to make sure it makes sense. FlowSum is a really great one to use. This is another clustering method. What I like to do is run a t-SNE and a FlowSum on the same merged file. I'll overlay my FlowSum onto my t-SNE, You can kind of see that in the middle there where we have like a color-coded t-SNE. If these clusters are broadly agreeing with each other, I can feel more confident that my t-SNE is actually showing me something true about my high-dimensional data. And we can do the same thing with traditional gating. You should do your same traditional gating, finding out your subsets of interest. And again, if these broadly agree with your clusters, you can feel more confident that they are real. Once you start to get to clusters that don't seem to conform to any known population, now you maybe found something interesting and you should start to dig into that. We can do that with heat maps, color axes, probe gates, just showing a few examples here. The color axes we can see on the far right here, just showing which clusters most highly express which markers I'm interested in. And this can start to give us an idea of which clusters correspond to which populations. We can also use a probe gate, just a small little gate we'll drag around our t-SNE that's hooked up to some histograms. And again, we can use that as a way just to identify our clusters and start to narrow down the ones that we know for sure what they are, and then find the interesting ones from there. And then just one last note on comparison. The main thing I like to look for is confirming that I see the expected clusters, that I'm not over clustering or something, and then looking for those novel clusters and trying to figure out what they are, making sure it's not noise, making sure it's not debris. But if it is real cells, then I can start to dig in on those and see if they are interesting to me or not. If we have different samples that overrepresent or underrepresent certain clusters, that can start to tell us things about what this condition or what this tissue is, how it's different from our other samples. And again, using those heat maps, those color axes, Flojo has this really nice feature now called Cluster Explorer that'll kind of open a lot of this for you at once. Just really digging into those clusters, finding what the identity of them is, and if they're relevant to your actual experimental question. So that is all I have for today, for all this time. Thank you, everybody, for your time. And if there's any questions, I'll take those now.
All right. Well, thank you so much, Bert, for that, very simplistic talk on a very complicated topic. I've been trying to figure out exactly what t-SNE is, so it was really nice listening to your talk. So thank you so much for sharing that. We do have some great questions coming in. So I want to start off with this one. So there's someone asking, is there some dimensionality of flow data where t-SNE might becomes more beneficial or even crucial for analysis? And then on the flip side, when would you caution a researcher away from using t-SNE?
I would say t-SNE is pretty low risk in that case, because we're never just going to run a t-SNE and then just take that as granted. Like I said earlier, we're not actually going to take that and send it off to be published. What's really important is that no matter how dimensional or how big or complex your data set is, you're taking that output and you're actually digging in to find out the identity of your clusters and things like that. In terms of when I would steer people away from it, if you're looking at less than, just to pick a number, 10 markers, t-SNE is probably not going to tell you too much. But again, there's no real risk in running it and just seeing what it does. There's not really, as long as you're not just taking it at face value, I don't think there's much of a risk of it leading you astray.
Right. So our next question's asking, is there a maximum number of antibodies that are appropriate for reliable t-SNE data?
No, I would say that it pretty much as high as spectral or even mass cytometry can go a little bit higher. Those are perfectly fine to use for t-SNE. It's really just going to be the more parameters you use, you might need to increase your iterations or your perplexity, and it's just going to increase the amount of computation time. But if you have the patience, if you have the computational power for it, it can really take as much data as you can give it. I will say, actually, as one quick note, not so much in terms of number of parameters, but if you just throw all of your data into a t-SNE with these millions and millions of events, you're really just not going to get very good separation. So downsampling is going to be really important for them.
Great, thank you. So there's a question from Drew. He's asking, do lower similarity groups tend to be less abundant cell types, so like basophils in blood?
When you say low similarity, that's always a relative measure. So just using basophils for an example, they're going to have high similarity to each other. But in terms of having low similarity, so if we looked at like the red, blue, and green from earlier, those would each be thought of as different cell types. But in terms of, I guess in terms of low similarity within a cluster, that's why perplexity can be important. If we have some clusters that are very tight in high dimensions and some that are very loose, having an appropriately high perplexity will make sure that they still do get clustered together in that final t-SNE output.
There's a question from Shannon. This is actually a question I also had. So is tSNE a type of PCA or just one that or just that it uses the T distribution?
It is somewhat similar to PCA or principal component analysis. I would say that the main difference is that with PCA, we're typically looking for the parameters or a few parameters that are explaining the majority of difference within our data. tSNE does not particularly care about which components are principal, for example. It's kind of weighing them all equally and is just looking for ways to cluster them based on that high dimensional similarity. So I say they are similar, but they are distinct in terms of what they are trying to tell us about our data.
Gotcha. So there is another question asking about whether there are, tutorials online to learn how to install these plugins and how to use them for, FlowJo.
Sure. The FlowJo Exchange, where you can download all of these, they do all come with a PDF telling you how to actually install them. If you're just patient with it, then you can get through it. I find almost always the stumbling block is people trying to install R and point FlowJo to the right directory. There are definitely tutorials on that. I'm sure if you type tSNE FlowJo into YouTube, you'll find plenty of step-by-step walkthroughs for it. One main thing I will say, whenever you're trying to run FlowJo or tSNE rather in FlowJo, bring all of your files local, just put them onto your desktop. I don't particularly know why, but I find that whenever I try to run from data that's on a network drive or something, I often get these crashes or just file not found. So just bring them all local would be one thing.
So you might have some insight on this in terms of when you're submitting for publication. Do you tend to include all graphs in the supplementary data?
I would say there's no real downside to it. If you're going to be putting in the supplementary data, I think it's always best to include more. Sorry. I'll say it's always better to include more in that case. But if you're just throwing in every possible tSNE you could think of showing with every possible heat map, I don't know. I try to focus down on the ones that are actually showing the story about my data that I want to tell.
So there's a question from Samuel asking, so for the computational analysis, do you use RStudio or can you do this analysis with FlowJo directly?
I just use FlowJo directly. I will fully admit that I'm not the best versed in R. FlowJo is just going to use R on the back end for it, so the output should not be appreciably different. And I just find that interface to be a little more intuitive. It's just what I'm used to also.
Okay, great. And then we have one more question. I noticed that there are some questions specifically asking about particular, experiments. I'm sure Bert would be happy to help you guys with troubleshooting if he can. So for those kind of specific questions, if you want to email Bert directly, I'm sure he'd be happy to help you out.
Sure.
But in terms of questions we're going to address here during the webinar, just one more question. There was someone who's asking if you could expand a little bit more on learning rate. They were a little confused by the concept of it.
Sure. So let me go back a couple of slides here. So when we're looking at on the left here, our low dimensional distributions, when we're doing these iterations and we're jumping from one to the next, the movement of our data points is again based on that high dimensional similarity. Similar points are going to attract and dissimilar points are going to repel. Learning rate essentially is how big are these jumps between the iterations. So the larger it is, we're going to have these big jumps, and the smaller is going to give us these smaller fine-tuning jumps. It's kind of similar to the early exaggeration I mentioned. Again, I think 7% of your total event is like what FlowJo recommends. I usually find somewhere in that 7 to 10% range. So if I have 1 million cells, 7 to 10% of that is my learning rate. Is usually a good place to start. It is one of the less intuitive concepts there, but it's essentially just, how brave is the algorithm feeling when it's taking these steps? Is it going to do big swings or just small little fine tuning movements?
Great. Thanks for clarifying. And if there are any concepts, after the workshop is over that you guys are still confused about, you can email us at Proteintech. You can also email Bert directly, and we will, find a way to answer your question for you. So with the last few minutes that we have, I just wanted to share a few more slides, just a few product slides that might be interesting for our audience today. So I'm going to go ahead and share my screen here. Okay. So I just want to share a few products that might be interesting for our audience. So the first products that I want to highlight are our correlate conjugated primary antibodies. So Proteintech offers primary antibodies that are conjugated to our correlate plus dyes. And our correlate plus dyes cover a wide range of wavelengths for easy multiplex panel design. They also have unmatched brightness and photostability, and they're equivalent to Alexa Fluor in their spectral profile. And all our conjugated antibodies undergo very stringent validation, so you can be confident that you'll obtain reliable results. And because of their quality and their performance, they're really perfect for your precious samples. Proteintech also offers antibodies that are conjugated to more of the conventional dyes for flow cytometry. So these include FITC, PE, and APC. We also offer Atlantic blue, which is spectrally similar to Pacific blue, and then Cardinal red, which is spectrally similar to Alexa Fluor 594 and Texas red. I also want to highlight our flexible antibody labeling kits as well. One of the challenges of flow cytometry or, many other antibody applications is you're finding a pre-conjugated antibody that you like, or in the case of flow cytometry, that fits your panel. So Proteintech offers a solution for that with our flexible antibody labeling kits. So Flexible is a novel antibody labeling kit that uses an affinity linker to conjugate fluorochromes, enzymes, and molecules in any buffer condition. So the protocol for Flexible is really easy. It only takes 10 minutes to do. So within 10 minutes, you can label your primary antibody and use it for your experiment. And this is regardless of the supplier of your primary antibody. So you don't necessarily have to use a Proteintech primary antibody in order to use the Flexible antibody labeling kit. And because you're pre-conjugating the antibody ahead of time, it's perfect if you're looking to use antibodies from the same species in an experiment. So you can use multiple mouse antibodies in one experiment if needed. Also, because it's simple and fast to use, Flexible allows you to easily rearrange your panel and customize your experiments. I also want to highlight a new product that was launched, which is our flow cytometry panels. So our flow panels are perfect for those who are, just beginning with flow, or if you're looking for just the convenience of a pre-designed and pre-optimized panel to help find your target cells. So our panels use top-sided clones that you know and trust, and they include 5 antibodies conjugated to different fluorophores. So what we do is we keep the PE channel open in these panels so that you can put in your target of interest. So currently we have these 6 kits that are on the bottom left, which have been designed and optimized by our experts in-house. And I also want to share a current promotion that we have going on, which allows you to try both our a flow cytometry panel as well as Flexible. So with this promotion, if you buy one flow cytometry panel, you can get a 10 reaction size of Correlite Plus 555 flexible antibody labeling kit for free. And so because the PE channel is open in our panels, Flexible 555 really fits in like a glove. So all you have to do is use the promo code FLXPNL23 to claim the offer, and we can provide this promo code in a follow-up email if you're interested in trying it out and using this promotion. And then finally, I just want to share that Proteintech also offers other supporting reagents and kits for flow cytometry, including isotype controls, secondary antibodies, viability dyes, cell health assays, and fix and perm buffers. And as an attendee of today's webinar, we'll be sending a follow-up email, which will have a link where you can choose one free full-size flow antibody conjugated to our Corelight Plus dyes from this list here on the right. So we'll also provide this list for you so you guys can take your time choosing which one you want to try out. We'll also provide a certificate of attendance as well as the recording of this webinar. So please do allow a week for this email to be sent to your inbox. So with that, I would like to thank everyone so much for joining our webinar today. Again, a big thank you to Bert for sharing all his knowledge with us and for simplifying tSNE for us. And if you do have any questions after this webinar is over, please do feel free to email us at events@ptglab.com, or you can email Bert directly at rlad@luc.edu. If you liked today's workshop and want to learn more about our upcoming events, please do follow us on our social media channels because we do post about our upcoming webinars and workshops. So with that, I would just like to thank everyone once again for joining our workshop today, and I hope you all have a great rest of your day. Take care.